So let's have a frank discussion, you and I. Let's talk about the birds and the bees, okay? When we talk about fertility traits, so often we hear people talking about it as a low heritability trait, okay? We hear this time and time again. And I think there's some consequences from how we keep framing fertility as a low heritability trait. So if we go to the American Angus Association website and look at their published heritabilities, for their heifer pregnancy EPD, they publish a heritability of 14%. So what that means is that 14% of the variation in heifer pregnancy is due to genetics, okay? With the other portion being due to environment or other factors. Let's put this in a frame of reference for a second. If we also look at the weaning weight EPD, the heritability for it is 20%, and the heritability for milk is 14%. So do we hear people running around all the time talking about weaning weight or milk as lowly heritable traits? I don't hear many people talking about growth or milk as a lowly heritable trait. And so I think this really can change our perspective about selecting for fertility. This is a graph from a publication of ours from the University of Missouri. And what we have here is the birth date of the animal, here on the x-axis. And here we have a measure of their genetic merit for milk production. And as you can see, due to selection decisions of Angus breeders, the genetic merit for milk has been increasing over time, okay? And so once again, and in fact, it's not only increasing, but it was one of the most strongly selected traits in the breed during that time period, from the 1960s to the early 2000s. If we look at heifer pregnancy, and this is a smaller number of bulls and a smaller number of cattle, and the heritability here is about the same, but instead of it going down, we see this slightly downward trend in heifer fertility over time. And I think perhaps that's because in producers' mind is this mindset that fertility is lowly heritable, and so we don't need to select for it. We can also look at the Hereford Association. They have a heifer calving rate EPD that they are in the process of deploying, and it has a heritability of 27%. So a fairly good chunk of the variation in that trait is due to genetics in the Hereford population. So I think as a consequence of this mindset, we've given a lot of cows and a lot of heifers a get-out-of-jail-free card, right? There's a lot of cows who get a second chance, a third chance, a fourth chance to get it right, whereas that if that heifer or cow had failed on one of our other traits, she would have been down the road. So let's talk about selecting for fertile heifers. One of the best ways to do this is, of course, through EPDs. Whenever an EPD is available, that's the right way to select for that trait. Here's some of the various breed associations and their EPDs. This table is in your handout, so I won't spend much time on it. So this is for the heifer's success in reproduction. We also have EPDs for the cow's repeated success in reproduction, and we have a 30-month pregnancy EPD. That is, given that the heifer calved as a 2-year-old, what is the probability that she also calved as a 4-year-old? And then we have some stayability EPDs and a sustained cow fertility EPD. So in some situations, we don't have EPDs for the trait that we want to measure, or we have a commercial population where we don't have EPDs on those animals. So whenever we see an animal that is open, that's something we don't want to see. We don't want to see open cows. And so a lot of times, one of the simple ways we can make selection is simply by sending those open cows down the road. In Missouri, we have an extension program. It's called the Show Me Select Heifer Replacement Heifer Program. And if you Google that, you can find more information about it, and there's also a video available. In this program, we collect a lot of information on the health of these animals and a lot of information on the reproduction of these animals. So we do a pre-breeding examination, we do the breeding, and then we do a pregnancy evaluation. When we do these pregnancy tests, a lot of times in this program, what we've moved to is using ultrasound to more accurately and precisely define how old that pregnancy is and various things like that. We also do what we call a reproductive tract score. This is a score where the heifer gets a score from 1 to 5, with 1 that heifer is a long ways off from reaching puberty, to 5 that heifer has reached puberty and now has a large CL follicle. And there's various endocrine events that happen during this process from prepubertal to pubertal. But the thing that gets me interested about this collection of data is that these heifers that score a 1 very rarely actually get pregnant. And these heifers that score a 5 are much more likely to get pregnant in this process. So this scoring seems to be predictive of whether or not that heifer is going to get pregnant in the long run. I'll talk a little bit about the research that we have going on right now. So in the project that Matt wanted me to talk about, what we've done is we've sequenced about 250 different bulls from 9 different breeds. And what we're doing when we sequence the entire DNA of these animals is we're looking for what we sometimes refer to as broken genes. Okay? So this is a gene that carries a variant that the effect of that variant is so severe that if you inherit 2 copies of that variant as an embryo, that embryo is going to be lost or aborted sometime during the pregnancy. Okay? And so these variants have a strong impact on the early pregnancy losses that we see in cattle populations. So when we sequence these 250 bulls, we'll find a large number of these variants that may or may not be breaking these proteins, breaking these genes. So then we have to have a way to go back and sort through these variants to see if we do in fact have these genes that are causing embryonic losses, embryo losses. And so the second part of this project is we then go and genotype 10,000 cattle to measure the frequency of these variants. And if the variant is at a certain level in a population, we should be seeing animals with 2 copies of that variant, right? So if that variant is compatible with life, we should see animals that have 2 copies of it. If that variant is not compatible with life, we'll never observe a heifer or a bull that has 2 copies of that variant. And so with that follow-up part, we can sort through to see whether or not we do have some of these variants that are causing embryonic losses. Once we find these variants that cause embryonic losses, we can then create a new chip, a new SNP chip, and we can test animals with this, and we can do things to manage those variants that are causing pregnancy losses. So one of the ways we can do that is through mate selection software. So we give the software the cow's genotype for the various embryonic loss genes. And then we give it a list of bulls that we're thinking about mating that cow to. And that software will optimize that mating to decrease the probability and chance of creating a pregnancy which won't be viable and will be aborted or lost sometime during the pregnancy. The other thing that we can do with that genotype information is just to create an EPD for pregnancy loss, okay? And so based on how many variants that animal carries that are broken genes, we can give that bull or cow an EPD score for pregnancy loss. One of the things that we do in animal breeding is find indicator traits, right? So one of the things that we routinely ask producers to do is to record birth weights. So is anyone actually getting paid for a birth weight? No, right? There's no economic impact of that birth weight. But that birth weight is a very strong indicator of whether or not that pregnancy is going to have calving problems, right? If that animal, the larger that calf is, the more likely we're going to have a calving problem. So birth weight is a very strong indicator of the economic trait of calving ease. We can do that same thing with reproductive traits, is to find indicator traits that help us do a better job of predicting fertility, okay? And so I talked about this Show Me Select Heifer program, and we did a little bit of pedigree analysis to measure how much of the variation in these traits is due to genetics. And so we looked at 1,500 commercial Angus heifers that were out of 180 different Angus bulls, and we pulled back the pedigree on those bulls. This reproductive tract score, which we take 4 to 6 weeks prior to breeding, and is an indicator of puberty status, is a really repeatable and accurate measure of the puberty status in those heifers. So when we do this pedigree analysis, we estimate that about 26% of the variation in this reproductive tract score is due to genetics. And so that lines up pretty well with previous publications of 32% of the variation in reproductive tract score being due to genetics. So some of the indicator traits that we're working on is things like reproductive tract score, pelvic measurements, and ultrasound pregnancy diagnosis to see if these are better indicators of fertility EPDs. And we're working on some BIF guidelines to incorporate and to kind of classify the best practices to measure these traits. The other thing that we can do and that we've proposed to do is look at the genomics of these indicator traits. So it's easier to record the phenotype on an indicator trait, and so we can do that for a large number of animals. I talked about previously in that embryonic loss sequencing project that we were going to genotype 10,000 animals. Well, 7,000 of those animals in that fertility project are going to have measurements for these indicator traits of reproductive tract score, pelvic area, and ultrasound pregnancy checks. And so we can do the typical genomic analyses that we've talked about today to look at the regions of the genomes and the genes that are predicting the variation in these fertility traits. And so we can use that functional chip that's been designed at the University of Missouri to look at these different things. And so with that, I'd be happy to take any questions. I know I very briefly covered the fertility sequencing project, so if you have specific questions on that, I'd be happy to answer any of those. I'd also like to point you to 2 web resources that we have. The first is eBEEF.org. That's a group of 6 of us, beef specialists throughout the nation that are working together to really get some good content and information about best practices in genetics and the new changes that we're seeing in genomics. And I'd also point you to my blog where I try to keep you all up to date on things that are happening in the industry. So with that, I'll take any questions.